जस्ट हो रहा है सर, हो गया सर।
ठीक है। Hello, welcome to this amazing platform of PW NEET English. Myself Biswajit, your zoology mentor in this particular channel. So if you want a complete, compact and rapid revision of this unit biotechnology, where we'll be learning two chapters. One is principle and processes and another is biotechnology and its application. Now every one of you is doing really great in your life and preparing for NEET 2026. So very good evening Pravakaran and all who are watching. Okay, so have you already completed these two chapters? Those who are watching this live session, this biotechnology principle and processes and the application, or you are watching for the first time? Have you already studied these two chapters, or came here to revise it again, or you want to learn for the first time? Can you please write down? Okay, so what do you understand by the word biotechnology? Obviously, it's a combination of biology with some modern technological aspect. Now, we can talk about old biotechnology or classical biotechnology as well as modern day biotechnology. What do you mean by old or classical biotechnology? It's the fermentation technology, the technology we use to produce curd, wine, bread. And for modern day biotechnology, we talk about recombinant DNA technology, test tube baby and all. Okay, so you are watching this for second time. No, I was asking that are you completed like this particular chapters for your upcoming exam? Have you already studied? Have you already learned this? So basically this session gonna be a rapid revision. Okay, so if you check your NCERT, the beginning of this particular unit, you're gonna get the name of two scientists. One is Herbert Boyer and another Sir Stanley Quinn. Okay, now they are pioneer of rDNA technology or recombinant DNA technology. They taught us how to construct a recombinant DNA and to put that or transform that inside another host cell. Let's talk about Herbert Boyer. So by 1969 he performed studies on couple of restriction enzymes of E. coli bacterium with especially useful properties. Now Herbert Boyer, he was expert of restriction endonuclease enzyme, which will be needing as molecular scissor to cut the DNA from specific segment. Yes. Inchara sir, hello, very good evening to you. Hello Chaitanya, good evening again. Okay, so let's talk about Herbert Boyer. So he was expert of restriction endonuclease enzyme, and you can also call those enzymes as molecular scissor. Like we use scissor to cut anything, to cut paper, to cut ropes. Similarly, biotechnologists are using restriction endonuclease enzymes. These are particularly kind of hydrolase enzyme, and we are using it to cut DNA molecule. Okay, so Boyer observed that these enzymes have the capability of cutting DNA strands in a particular fashion, which left what has become known as sticky ends on the strands. Now, these clipped ends made pasting together pieces of two different DNA a simpler exercise. Now, what do you mean by this? if you take a, a double-stranded DNA segment, then they obviously they are linked with base pairs, right? Now, if you want to separate them, you need that particular molecular scissor, and that molecular scissor will be inspecting the codes written on this DNA, and after checking, they will make proper sticky end cut in between some specific region, and the resultant pieces will be getting like this. So this kind of ends are called as sticky end cut. Now, what is the contribution of Stanley Cohen? Cohen had been studying small ringlets of DNA called plasmid. Yes, inside bacterial cells, you will be finding a self-replicating double-stranded circular DNA which are extra chromosomal in nature, and we call them plasmids, which float about freely into the cytoplasm of certain bacterial cells and replicate independently. Mean they don't need help of nucleus or nuclear of the cell from the coding strands of DNA. So Cohen had developed a method of removing these plasmids from the cell and then reinserting them in other cell. So Stanley Cohen, he was master of plasmid. Herbert Boyer, he was master of restriction endonuclease enzyme. And when they came together, they built something amazing. They built the first example, the first recombinant DNA for us. Okay. Now, if we talk about biotechnology, so biotechnology deals with the techniques using living live organisms or the enzymes of those organisms. And why we are utilizing these live organisms and their enzyme? We want to produce some products and we want to use them for some processes, and obviously those products or processes will be helping us for upgradation of our lifestyle. If we talk about products, they are talking about proteins, enzymes, newly formed genetically modified organisms. If we talk about processes, we got PCR technique, polymerase chain reaction, we got gel electrophoresis technique, we got autoradiography technique, and many more. Okay. Now, if we talk about classical biotechnology, all of them were microbe mediated process. Now, everyday life in your kitchen or in your home also you can perform that. You can produce curd, you can produce wine, or you can produce bread or beer, right? So this is called fermentation industry. Okay, not an issue, just the beginning of the class. What's your name actually? You are commenting as QTPie124. Okay, not an issue. So these are some products of classical biotechnology. This is curd, this is bread, this is wine. And remember, this all these things will be coming under fermentation technology. Okay, the old biotechnology. Now we'll talk about modern day biotechnology or today biotechnology. So for modern day biotechnology, we talk about in vitro fertilization. That means fertilization outside the female reproductive system, and thus test tube baby program is there. We already studied that we need the help of test tube baby program in art process, right? If you observed, if you studied the chapter reproductive health, the last topic is assisted reproductive technologies. Means infertile couple, right? if they want to get baby, they have to take help of biotechnology through in vitro fertilization. Now, synthesis of using a gene, then preparation of DNA vaccine, right? Like using recombinant DNA technology, hepatitis B vaccine was produced, and correcting a defective gene, we call it gene therapy. Okay, got it. Your name is Sapna. Okay, so these technologies are part of modern day biotechnology. These are some instance or examples: test tube baby, formation of uh, recombinant gene, preparation of DNA vaccine, and gene therapy. Right. Now, European Federation of Biotechnology, full form of EFB is European Federation of Biotechnology, has tried to merge the definition of classical biotechnology and modern biotechnology in this manner. So, traditional view plus modern molecular biotech is punched here, while the EFB describe the definition or try to define the biotechnology. So, according to EFB, it's the integration of natural science, that means physics, chemistry and biology, and organism, cells, parts thereof, and their molecular analogues. They are talking about cells, cell organelles, DNA, RNA, protein, carbohydrates, lipids for the products and the service. Okay, what kind of service? Like test tube baby, right? Genetic modification, formation of GMO, genetically modified organism, cloning, right? So these will be coming under service, or gene therapy will be coming under services. Now, product means refined enzymes, then refined proteins. These are examples of products. Now, what are principles of biotechnology? So, there are two core techniques are considered as main two principles. What are they? First, genetic engineering. Second, bioprocess engineering. Now, what do you mean by genetic engineering? Is the method of altering the sequence of DNA or RNA. So, here we will be manipulating genetic material. The process of manipulation of DNA and RNA, here there will be alteration of the chemistry of DNA and RNA, inserting that recombinant DNA, that altered DNA, into a host organism to obtain the product, and change in phenotypic expression in the host, right? Now, in a story format, if I want to say that, if I want to explain that, you are a normal human being. Now, you have observed Spider-Man, though Spider-Man is a hypothetical being, right? But suppose one day biotechnology will get some advancement, and it's in the laboratory. Now, in near future, it will be quite possible to make a Spider-Man. So, what you have to do? You have to extract the spider web forming gene from the spider, and you have to incorporate that gene within the genome, within the DNA of human being, and in this manner you can produce a real Spider-Man. Though it's hypothetical, but if we want to convey the what is the actual motto of biotechnology, that to make a normal organism superior, or a normal organism to work for us. Like inside our body, our pancreas is producing insulin hormone, which is required to low down glucose level in our body, right? But from nature, it's not possible to obtain human insulin. But biologists or biotechnologists made this possible. How? They took the particular piece of DNA from human genome, which can produce human insulin, and they inserted into the bacteria called Escherichia coli or E. coli. Now this E. coli is transformed by inserting human DNA. Now this transformed E. coli or genetically modified E. coli can produce exact insulin which is useful for us. Okay, so that is all about genetic engineering. Now what is the second principle? That is bioprocess engineering or biochemical engineering. Now what is the importance of bioprocess engineering or biochemical engineering? Now genetic engineering is a kind of lab work. It's a trial period. You have made something superior or something good for the humanity, right? But now the next step will be to market that. Now you want to sell that product into market, right? You need to produce thousands or millions of copies of that. For that you need a specialized laboratory, right? So maintenance of sterile ambience for the growth of those genetically modified prokaryotes or eukaryotic cells to obtain enzyme, hormones, antibiotics, drugs and proteins. So these are the things which can be done under bioprocess engineering. With an example I am explaining it. Now when the COVID-19 was affecting the humanity, now in different countries, different scientific or biotechnological institutions, they started researching on that particular virus, how to produce how to produce vaccine, and some of them or few of them produced DNA vaccine or recombinant DNA vaccine, which is quite effective against that COVID-19 virus. So that was a kind of invention by them. So that falls under genetic engineering. Now what is actual need of that particular invention? You have to sell to the market, you have to give to the hospitals, and those hospitals will be injecting those vaccine against COVID-19 into human being. Now for that you have to create millions or crores of copies of that DNA vaccine which was prepared in the lab. Now for that you need a huge facility, and that should be sterilized. That ambience, you have to provide that ambience, and providing that particular ambience to produce a genetically modified organism or to culture it or to obtain the product is biochemical engineering or bioprocess engineering. Okay. Now why we need to opt for biotechnology? Why do we need to opt for biotechnology? Right. When you talk about food science, the maximum utility of biotechnology is in agriculture sector and in medicine sector, right? Now if you want to implement biotechnology into food science or agricultural science, many scientists will say we already can perform hybridization. We have hybridization technique, and by using that we can produce superior varieties of crops and the economic animals. Now against that you have to say that no, the traditional hybridization technique they are using to produce superior variety of crop or economic animals, they have certain limitation. Rather, if we use biotechnology, the process will be more rapid, more fast, and the products will be much more qualitative. Okay. Now, that's why NCERT has given this topic. What is the advantage of sexual reproduction? First, we need to know about what is the advantage of sexual reproduction over asexual reproduction. Now, it's very clear. Asexual reproduction will be conserving genetic information, whereas sexual reproduction will be permitting variation. And that's the reason different varieties or hybridization can be possible by following this sexual mode of reproduction, whereas if you are following asexual mode of reproduction, identical twins will be produced as the progeny. So obviously we will be omitting asexual reproduction, we will go with sexual reproduction. Now again we were debating that even biotechnology is surpassing sexual mode of reproduction in terms of effectiveness. So the next topic is advantage of genetic engineering over the traditional hybridization procedure. Now what is the naive of traditional hybridization procedure? Is the sexual mode of reproduction between two superior varieties. Like if you want to give one of the example, like in India we have some native varieties of cows, like you take Sahiwal, a good variety of Indian cow, right? Now that Sahiwal cow in a single day can produce maximum 7 to 8 liters of milk. Now that is a native Indian cow. Now if you check, if you compare with the Jersey cow, which is not present, which is not native of India, right, which is a native cow of America or USA. So the capability or lactation capability of Jersey cow is near about 30 to 35 liters of milk in a day, in a single day. So you can check the difference. So as a farmer, what you want to bring the Jersey cow in India, right, into your cow shed, into your cow farm, but unfortunately they cannot survive in this environment, in this climate. Now you need to take help of a biologist. So he or she will be suggesting you perform hybridization between a Jersey bull and Indian Sahiwal cow, and the offspring or the progeny will contain gene of Jersey and gene of Sahiwal, and that hybrid variety of cow is called as Jersey Sahiwal. But still that hybrid Jersey Sahiwal cannot lactate like its parental generation, right? The hybrid Jersey Sahiwal can only produce 15 to 20 liters of milk, okay, as because though it got some good genes from both of their parents, both of their father and mother, but some bad genes also were inherited during the time of their hybridization, and that is the drawback. When normal sexual reproduction will be happening or hybridization will be happening, along with good genes, some bad genes will also be inheriting into the new progeny. Now you are very much target specific. You want only the good genes to be inherited to the next progeny. So in that case you have to omit the traditional hybridization technique and you need the help of biotechnology, right? And how biotechnology will be helping you? Biotechnology will be helping you to select the gene of interest from the donor organism and to insert that gene of interest successfully into the host organism, which is gonna produce the product for you, for your welfare, for human welfare. So that is the charisma of biotechnology or genetic engineering. So it's quite easy. Hybridization, if we perform hybridization along with good genes, some bad genes also be inherited, but if you are performing rDNA technology, recombinant DNA technology or biotechnology or genetic engineering, then you can only cut the desirable gene or the good genes from the donor organism, and you can culture that particular gene into the host organism to produce your desirable product. Okay. Now two columns are given and we have to match them. some techniques are written here: hybridization, asexual reproduction, sexual reproduction, and rDNA technology, recombinant DNA technology. So, which of these will be permitting variation? Okay, sexual reproduction will be permitting variation. Which of these techniques will be used for inclusion of undesirable genes? Hybridization. If you are performing hybridization, the inheritance of good genes and bad genes will be there. Okay. Now, isolation of only desirable gene. If you want isolation of only desirable gene, then you have to perform recombinant DNA technology. And if you want to preserve the genetic information progeny by progeny, then only you have to go with asexual mode of reproduction. Okay. Now, the next question that is asked by NCERT, that is, do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Okay. Now, as you have studied today that you can perform genetic engineering rather performing hybridization technique. And what is genetic engineering? You have to select an organism, as because that organism is containing a special gene inside it, and the product of this particular gene, that is called as gene of interest, is useful for you. So what you have done, you have taken another organism which can be cultured into your laboratory, right? You have taken another organism that is called as host organism which can be cultured into your laboratory. So simply, without gaining full knowledge, you have extracted the gene of interest from the donor organism, right? And directly you have incorporated the gene of interest into the cytoplasm of host cell. Okay, then what will happen? As because this, for this host cell, the gene of interest is nothing but a foreign particle. So it will pose a immune attack and the host cell will be destroying the gene of interest into it if you are directly inserting that inside that host. So for that purpose you need to do something else. Okay. Now that is the answer of, do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? So if you are directly inserting the gene of interest into the new organism, then the gene of interest will simply die. The gene of interest will simply be destroyed by the alien organism or the host organism, and there is no probability that gene will be inherited to the next progeny. Now, for that, what you have to do? So, you got a new need. And what is the new need? You need to tag or incorporate the DNA with the genomic setup of the host organism. What you have to do? You have to incorporate the DNA with the genome of the host. So, while the host organism is performing the central dogma, that means inside the host cell, the DNA will be converted into messenger RNA, mRNA, and mRNA will produce protein with the help of ribosome. So this is called central dogma, and that is the importance of the genome of host. So what you have to do, you have to incorporate the gene of interest in directly into the genome or chromosome of host cell. Okay, rather just putting it into the cytoplasm of that alien cell or the host cell. So now what will happen when you are incorporated, when you have incorporated the desirable gene inside the chromosome or genome of the host, now that gene of interest will replicate or multiply inside the host and will be inherited to the next progeny. So again I am making it clear. Now this is the host cell and this is the gene of interest. This is the gene of interest and this is the genome of the host. This is the genome of the host. So you have to incorporate the gene of interest inside the genome, right? Now when this host cell will replicate, means will perform cell division, then the gene of interest will also be inherited with the replicated DNA, as because this gene of interest or the desirable gene, that is part with the by following the process of recombinant DNA technology, you have incorporated the gene of interest inside the host genome. Okay, or else you can take help of another thing. And what is that? Suppose this is another host cell, this is the genome or the main DNA or the nuclear DNA of the host cell, and you don't want that your gene of interest to get incorporated into the DNA or genome. So for that you have to search for these circular ringlets in case of bacterial cells, and we call these ringlets as plasmid. So another chance is there, if you incorporate the gene of interest inside those plasmids, then also the bacteria can transmit the gene of interest to the new cells or to the next progeny. Okay. Now we have to study the first instance of construction of an artificial recombinant DNA, and that was performed in 1972 by those two pioneers, Herbert Boyer and Stanley Quinn. Now I will be talking about the first artificial recombinant DNA which was constructed and transformed inside E. coli bacteria. Right. Now let's study the construction process of first recombinant DNA. Okay. So they observed a particular variety of a bacteria called Salmonella typhi, which is responsible for causing typhoid fever, right? Okay. Now inside that Salmonella typhi, they got an extra chromosomal circular DNA, and that was basically plasmid, and name of the plasmid was R plasmid. Okay. Now that R plasmid was containing a very special set of gene which is having the capability to inhibit the effect of the antibiotic tetracycline. Means they are kind of super Salmonella typhi. Normal Salmonella typhi will die if you apply tetracycline antibiotic on them. But this super Salmonella typhi, as because they have this tetracycline resistance gene present into their R plasmid, with the use of tetracycline they cannot be destroyed. Right. So that was their observation. Now what they wanted? They wanted to take out this tetracycline resistant gene from this plasmid of Salmonella typhi and to culture this within the E. coli bacteria inside the laboratory. Okay. Now already we have learned that directly you cannot insert this into the host bacteria. So for that they have chosen another strain of Salmonella, that is known as Salmonella Panama. Okay. Now this Salmonella Panama, another strain, another variety of Salmonella it was containing a plasmid inside it called as PSC plasmid. The Salmonella Panama was containing a plasmid inside that is known as PSC plasmids, plasmid of Stanley Cohen. Now they took out that plasmid from Salmonella Panama. They cut that plasmid with the help of restriction endonuclease or molecular scissor, and within this plasmid called PSC, they have incorporated the gene of interest, that is the tetracycline resistant gene, which was isolated from the R plasmid of Salmonella typhi inside that PSC plasmid. And that is how they had constructed the first artificial rDNA called as PSC 101, plasmid of Stanley Cohen 101. You need to remember this. The first artificial recombinant DNA which was produced by Boyer and Cohen, the name is PSC 101, plasmid by Stanley Cohen 101. Okay, again I am repeating, they found a beautiful gene inside Salmonella typhi, inside the plasmid of Salmonella typhi, and that gene is responsible to make the bacteria resistant against the antibiotic called as tetracycline. Okay, now they isolated the gene. They had isolated the gene, that is tetracycline resistant gene from Salmonella typhi, and inserted that piece of gene inside another plasmid obtained from Salmonella Panama called as PSC plasmid. And that is the example, or this was the instance they constructed the first recombinant DNA. Why we are calling this as recombinant DNA? This PSC 101, here the remaining portion of the plasmid is of this PSC from Salmonella Panama, but this TET-R part, this gene of interest part, is from the plasmid of Salmonella typhi. So here DNA of two organisms are punched together to form PSC 101. That's why we can call these as recombinant DNA. Two DNA are recombined. Now you can do the process. Now you can insert this PSC 101 plasmid inside the cytoplasm of our host bacteria, that is E. coli. E. coli is very much easy to culture inside the laboratory. Now whenever they had inserted that recombinant DNA inside the E. coli, it was quite possible, it was quite easy for them to produce antibiotic resistant or tetracycline resistant E. coli and its progeny, and thus it said that the E. coli bacteria is transformed when you successfully incorporate the recombinant DNA into the host organism, and the host organism can express the gene, the host organism can produce the product, then we say that host is already transformed by the presence of recombinant DNA. Okay. Now the next topic, that is basic steps of genetically modified organism or GMO. So you can hence infer that there are three basic steps in genetically modifying an organism. So what are those three steps? First, identification of the DNA for desirable genes, and we call that gene of interest, GOI. Next, introduction of the identified DNA into the host. Next, you have to transform the host. You have to put that gene of interest with the help of a vector inside the host organism. And the third step is maintenance of introduced DNA into the host and transfer of the DNA to its next progeny. That means when the host will be, if it's a bacteria, then when it will be doing cell division, when it will be doing binary fission, then the upcoming progeny, upcoming E. coli gonna pause that tetracycline resistant gene of interest inside them. Okay. Now we'll be talking about tools of recombinant DNA technology as given in NCERT. So the tools we need to talk about, the tools will be studying about restriction enzymes, ligase enzymes, gel electrophoresis technique, about cloning vectors, and how to make the host cell competent, and the process of insertion of DNA if you want to transform the host cell. Right. Let's start with restriction enzymes. In shortcut form, we call them REN, R-E-N, restriction endonuclease. Okay. So in the year of 1963, three scientists, Smith, Arbour and Nathan, inside the bacteria Escherichia coli or E. coli, they discovered two important enzymes, right? And what are those two important enzymes? First is responsible for methylation of its own DNA, and that enzyme is named as methyl transferase. And second enzyme that is having the capability to cleave or cut the viral DNA into pieces, and that particular enzyme is called as restriction endonuclease. So again I am repeating, in the year of 1963, Arbour, Nathan and Smith, they found two important enzymes working inside E. coli bacteria to counter the viral attack. Now which virus is responsible for attacking E. coli? It's bacteriophage. Yes, I will be completing the one short series of zoology. You can check other lectures. Okay. Now, what is the function of this methyl transferase enzyme? See, if I am the virus and if I am attacking the bacteria, if I am the bacteriophage virus and I will be attacking the bacteria, so what I will do? I will inject my genetic material inside the cytoplasm of that bacteria. Now gradually the cytoplasm will go and bind or be a part of the bacterial genome. Now the bacterial genome will start working for me, right? But the bacteria is very much clever. Like we have our immune system inside our body, immune cells like WBC, antibodies, we have bacteria, they have methyl transferase enzyme. So what they do, this methyl transferase enzyme, they will do methylation of their own DNA. They will change the genetic makeup, and when the methylation of DNA is done, the viral genetic material will not be able to be incorporated with the bacterial genome. Got it? So that was the importance of methyl transferase. And another is very clear, restriction endonuclease. Whenever the virus is injecting the genetic material inside the cytoplasm of that bacteria, that restriction endonuclease is cutting into pieces and destroying the viral genome. Okay. Max, you are asking, will you take the full zoology chapters from class 11th and 12th, because it was very useful. Yes, obviously Max. All the zoology chapters already in one shot manner available in our channel PW NEET English. You can check them. Even they are given as a live format. Okay. So let's focus on this restriction endonuclease enzyme. So if you want to classify them, there are total 6 classes of enzymes are there. There are total 6 classes of enzymes are there. Okay, and they are named as OTHLIL, oxidoreductase, transferase, hydrolase, uh, lyase, then isomerase and ligase. Total there are 6 classes of enzymes, 6 groups of enzyme, 6 classes of enzymes are there, and these restriction endonuclease, they belong to the class hydrolase. They belong to the class hydrolase. Okay. Now this hydrolase have a subgroup called as nuclease enzyme. Now nuclease enzymes are responsible for cutting RNA as well as DNA. Nuclease enzyme, they are responsible for cutting nucleic acid. Okay. Now this nuclease enzyme can be of two types, exonuclease and endonuclease. Now what is the difference between exonuclease? Exonuclease enzyme will cut DNA from the edge, from the edges, from outside, and the endonuclease will be cutting the DNA from inside. So that's why we are calling them endonuclease. And this restriction endonuclease, they comes under this endonuclease kind of enzyme, right? Now we will be detailing this endonuclease enzyme. Now how they will be working, how the endonuclease enzyme will be cutting the piece of DNA. They will first inspect throughout the length of DNA. If this is the restriction endonuclease, it will check, it will run or walk through the length of DNA molecule, and it will be searching for a special sequence, and we call it recognition sequence. It will be searching for a special sequence. We will be calling this as a recognition sequence, and most of the time these recognition sequence are palindromic in nature. Now what do you mean by palindromic? Like if you read this terminology, this word Malayalam, from here Malayam. Now if you read it into reverse direction, it will be the similar Malayam. Madam, Madam. Forward direction also madam, backward direction also madam. Dad, forward direction also dad, backward direction also dad. So this kind of words are called as palindrome or palindromic sequence. Now similar kind of thing in case of genetic sequencing, if you are getting inside the DNA, those sequences are called as palindromic sequence. Now here for E. coli bacteria, there is a restriction enzyme called EcoR1, and for EcoR1 the restriction side or the palindromic side is like this, GAATTC, from 5' to 3' direction. Now if you read it into 3' to 5' direction, from yes 3' to 5', the reverse, the anti-parallel segment, here also you will be getting GAATTC. So from 5' to 3' it's GAATTC, from 3' to 5' also it's GAATTC, and that's why it's forming a palindromic sequence here. And if the restriction enzyme is EcoR1, it can cut the DNA by searching when it is getting this particular palindrome, and exactly it will put cut in between G and A on 5' to 3' direction, and 3' to 5' direction also it will put a cut on in between G and A, and thus it will be separating two strands of DNA. Now if we talk about another restriction enzyme, HIND2. Remember, this is the first discovered restriction enzyme, HIND2, and the palindromic sequence this will be searching for, that is GTTAAC. Okay. Now we'll talk about different types of restriction enzymes, different types. So there are mainly two types of restriction enzymes are there, and generally for biotechnology we use type 2 restriction enzyme. Okay. So what about type 1 restriction enzyme? They have dual function. They can perform the work of methylation. They can cut the DNA piece. So we already talked about one methyl transferase enzyme and another is restriction endonuclease enzyme. But if you are taking type 1 restriction enzyme, they are less specific. Type 1 restriction enzymes are less specific and they can perform both the work. But here we want someone which can perform some specialized work, and for that we have to choose type 2 restriction enzyme because they can only produce cut. They will not waste their energy by doing methylation of DNA. They are responsible, their whole and sole purpose is to provide cut into specific sequence of DNA. Okay, that's why they are used in genetic engineering, this type 2 restriction enzymes, and they can produce either sticky end cut, they can sometimes produce blunt end cut. Now few restriction enzyme will be producing sticky end cut and few restriction enzyme will be producing blunt end cut. Like we as of now we have talked about two restriction enzyme, one is EcoR1 and another is Hind2. Now this EcoR1 can produce sticky end cut and Hind2 will be producing blunt end cut. Remember, okay. So for if some restriction enzyme is producing blunt end cut, that is not much usable for us if you want to construct a recombinant DNA. So that's why most of the time we will be choosing those restriction enzyme who are responsible for producing sticky end or flanking end cut if you want to perform rDNA technology inside your laboratory. So this is how sticky end cuts will be helping you to form this kind of overhanging ends, and these overhanging ends are called as sticky end. And EcoR1 is a very good example of restriction enzyme that is widely used in genetic engineering laboratories. Okay. But you need to remember the name of Hind2. This was the first discovered restriction enzyme. Now you may ask, sir, why Hind1 is not first discovered enzyme? Okay, the property of Hind1, was understood much later then this Hind2. As a restriction enzyme, the property of Hind1 was understood much later than Hind2. That's why though Hind1 was discovered earlier as an enzyme, but as restriction enzyme, the property of Hind2 was understood for the first time. So that's why we regard Hind2 as the first discovered restriction endonuclease. Okay. And the restriction endonuclease which was used by Hubbard and Hubbard Boyer into the experiment, that was EcoR1. Now we'll talk about the nomenclature. Why restriction enzymes are given the name like Hind1, Hind2, EcoR1. Now let's talk about that. See, though in nature there are more than 900 restriction endonuclease enzymes are there, they are isolated from 230 different varieties of bacteria. So as of now, for biotechnological use, over 900 restriction endonuclease enzymes are isolated from more than 230 varieties of bacteria. And how they are named? Now if you check the name EcoR1 or Hind2, the first letter for EcoR1 is E and the first letter of Hind2 is H, right? So the first letter is the genus of the bacteria from which we are isolating them. For EcoR1, E means Escherichia coli, and for hind 2, H means Haemophilus. Okay, so isolated from the bacteria Haemophilus influenzae. So we have taken the first letter of Haemophilus. Now, what is the importance of second and third letter? Okay, the second and third letter is telling us about the specific name. Like, what is the full form of E. coli? Escherichia coli. So, what is the second and third letter? First two letter of the specific epithet. Escherichia coli, E. coli. So, CO is taken from here. Hind, Haemophilus influenzae is the specific epithet. So, second and third letter, that is in, that has been taken from influenzae word. Okay. Now, what is the importance of fourth letter? Now, fourth letter tells us about the strain of the bacteria. Like, the fourth letter for EcoR1 is R. So, R means that has taken from the RY strain of E. coli bacteria. Now, it is optional. You can use the fourth letter, and sometime the fourth letter cannot be used or can be omitted. Like, for example, uh, PVU1, PVU1. So, here we got first letter, second letter, third letter, PVU, and we do not have any fourth letter here, right? And the next thing, we will use a Roman number to denote the order of isolation. So, eco R1, this is the first order of isolation, hind 2, second order of isolation. So, that is the importance of the different signals or the different letters or numericals which are used to display the name of restriction enzyme. Okay. So, this slide is telling the similar kind of thing. If we check the name of EcoR1, E is taken from Escherichia, thus genus name. CO is taken from the specific name coli, COLI. So, CO is been taken. R is taken from the strain called RY13 strain. So, the first letter of R113 is R, and first eco R1, that is order of isolation, means while the scientists were checking for kind of restriction enzyme inside the E. coli, into their first trial they were able to isolate and identify this enzyme. That is why it is named as R1. Okay. Now, I was telling that there are few restriction enzyme where the fourth letter is omitted. See, for EcoR1 you have the fourth letter as R, but for SAL1 the fourth letter is absent. For PST1 the fourth letter is absent. PVU1 the fourth letter is absent. Okay. Now, the next enzyme you will be needing to perform rDNA technology, that is ligase enzyme. Now, ligase means enzyme which is necessary for ligation. Now, it is opposite to the lysis. Lysis means to break down. Restriction endonuclease, they belong to the class hydrolase. They can perform lysis, they break down, they cut the DNA, but ligase, they joins, right? They are responsible for joining to different pieces of DNA. Okay. So, when you need to construct a hybrid or recombinant DNA molecule by taking multiple pieces of smaller DNAs, then to connect them, to link them, you need to use ligase enzyme. Yes, and this ligase enzyme, they are responsible for formation of sugar phosphate backbone. They are actually making a phosphoester bond. Now, the process of separation of DNA according to their size. Now, you have isolated DNA from the host cell. Now, you need to learn about another process by utilizing which you can separate out DNA of different size. Okay. And the name of the process is gel electrophoresis. Suppose you have taken, you got a mixture of DNA where few DNA pieces are of 10 base pair, few DNA pieces are of 100 base pair, and few DNA pieces are of 1000 base pair. So, how you are gonna separate them based upon their size? Okay. So, for that you have to prepare a gel plate, and this gel plate, the gel is actually agarose gel, which is obtained from some seaweeds. They are red algae like Gelidium, Gracilaria. So, while we will be performing gel electrophoresis, the gel that is used is nothing but extract of red algae. Okay. Now, what you have to do? This is the gel electrophoresis machine and the gel plate. Towards the cathode, towards the negative electrode, you will be finding some cavity kind of structure. We call them DNA loading wells. So, that mixture of DNA is loaded inside these wells near to the negative electrode or near to the cathode. And what you have to do? You have to switch on the machine. You have to switch on the machine. Now, you know that negative electrode will repel DNA molecules, because DNA molecules are also negatively charged, right? Due to the presence of phosphate, DNA molecules are negatively charged. So, the opposite electrode, that is anode, the positive electrode or anode, gonna attract the DNA molecules towards it. So, gradually what you will be observing, the DNA molecules are moving towards the positive electrode or towards the anode, okay, through the pores of this agarose gel. Now, obviously, if there is a running competition, if someone is thin and fit, that person can run very fast, and some other competitor is very fat and weak, he or she cannot run really fast. So, in the case of DNA, the similar thing is happening. The larger pieces of DNA will remain into these particular zones, and the smaller pieces will be moving near to the anode. And this is how you can separate out different DNA of different base pair, okay, by checking their zone on agarose gel plate. Either they are towards anode or they are towards cathode. Towards cathode, these are the larger pieces of DNA. Towards the anode, these are the smaller pieces of DNA. Okay. One more question is asked: why agarose gel is used to separate out this DNA based upon their size? Because this agarose gel is having sieving property. It can filter out the DNA in course of their movement. Okay, it will be promoting movement of smaller DNA faster and larger DNA slower. Okay. Now, as because DNA molecules are very much minute, and under like even normal light you cannot observe them, but how to isolate? If you are not observing these DNA strands inside the agarose gel, it will be impossible for us to extract or isolate. So for that, what we have to do, for that we have to stain, we have to color this gel plate with the help of a special dye called ETBr, full form is ethidium bromide. Okay. Now this ethidium bromide is visible under ultraviolet rays. The ethidium bromide will go and bind with the DNA molecules, and when we will put ultraviolet ray, then it will give you a orange colored reflection. So if you are staining this dyeing this gel plate with the help of ethidium bromide stain, then only it will be visible to us under ultraviolet ray. Okay. Now once you have marked the gene of interest or the desirable pieces of the gene by the process of scooping, you have to take out the agarose, that portion of agarose gel, and by the process of elution, by utilizing elution buffer, special kind of buffer medium, we will be separating out and collecting the smaller DNA strands, if these smaller DNA strands are desirable DNA piece. Okay. Now we will talk about numerous cloning vectors. Now as we know that directly we cannot insert the gene of interest into the host cell. So for that we need some introducer. We need to introduce the gene of interest to the genome of the host cell, otherwise the gene of interest will die inside. So for that we need to take help of cloning vectors. Yes. So these cloning vectors are also called as gene taxi or vehicle or to deliver the gene of interest inside the host. Like we utilize cab service or we use car to move from one place to another place. Similarly, in biotechnology laboratories, these cloning vectors will be used as vehicle or the gene taxi to transform a normal cell with a recombinant or the gene of interest. Okay. Now, if you want to transform a bacteria, then we can use plasmid vector or we can use disarmed bacteriophages, right. Nowadays some different kinds of vectors are used, cosmids, phasmids, bacterial artificial chromosome, but these are not mentioned in NCERT. But as of now, you need to remember that the kind of cloning vectors which are used to transform a bacteria, either you have to use plasmid or you can use bacteriophages. Okay. Commonly plasmids are used to transform bacteria. Now what about plant cells? Okay. Now plants are of two types, dicot plants and monocot plants. Specifically, if the plant is dicot plant, they have a pathogen called a special bacteria called as Agrobacterium tumefaciens. So it's the normal case of Agrobacterium tumefaciens. Okay. So this Agrobacterium tumefaciens, that is having a special plasmid called as TI plasmid or tumor inducing plasmid. TI for tumor inducing plasmid, and that tumor inducing plasmid is having a special set of DNA that is called tDNA that can cause tumor or can cause cancer in plant. So that is normal function of Agrobacterium tumefaciens. But in lab, what we will be doing, we will be disarming the Agrobacterium tumefaciens, we will be taking out the TI plasmid, right, and we will cut tDNA, the harmful part which will cause cancer, and in the place of the tDNA we will be pushing, we will be inserting our gene of interest. Okay. So in that case, now when you are utilizing genetically engineered Agrobacterium tumefaciens and you will be infecting dicot plants, they will deliver the gene of interest inside the cells of these dicot plants. So that's the reason we say that these Agrobacterium tumefaciens can act as natural genetic engineer. It's because naturally this Agrobacterium tumefaciens is having the capability to transform a normal plant cell into a tumor causing or cancer causing plant cell. So what we have to do here, we just have to eliminate the cancer causing gene of plant, and instead of that we have to replace with our desirable gene or gene of interest. Okay. Now let's talk about plasmid. Now you know that plasmids are self-replicating extra-chromosomal circular double-stranded DNA ringlets. Now why they are extra-chromosomal? We know that for a bacteria there is only one chromosome, that is the nucleoid, right? Now if there is any other piece or other rings of DNA you are finding inside the cytoplasm, and they can produce their own copies. They can produce their own copies. So these ringlets will be called as self-replicating, and obviously they are circle. If you observe their shape, so they are circular. They have two strands, that's why they are double stranded circular rings. Okay. Now there can be two kinds of plasmid present into bacteria. One is high copy number plasmid, another is low copy number plasmid. Okay. Now how high copy number plasmid means they can produce 10 to 100 of their copies, and low copy number plasmid means they can produce 1 to 3 copies only. Okay. Now what makes this high copy number plasmids different from this low copy number? It's about the origin of replication. Okay. Now if you check the length of a DNA, and if you want DNA replication, so the place, so the point of that particular DNA from where the replication will be initiated or the replication will be started, that is called as origin of replication. Now if the origin of replication is easy to open, easy to unwind, then that particular plasmid will produce multiple number of the copies. If the origin of a DNA is very tough to open, then that plasmid will be creating very low number of the copies. Why it's so? Generally it is observed that the high copy number, for high copy number plasmids, the origin site of replication is containing more A and T. You know that when there will be base pairing in between the strands, A will bind with T and G will bind with C. Now A binds with 2 hydrogen bonds, G binds with 3 hydrogen bonds with C. Now if a DNA segment is containing more A and T, if a DNA segment is containing more A and T, then what will happen? There will be only double bonds. There will be only double bonds, and it is much easier to open double bonds in compared to triple bonds. So this is one piece of DNA. This is another piece of DNA. Right? Okay. Now here you are finding there is more G and C are present. So in between G and C there will be triple bonds, in between A and T there will be double bonds, and in between G and C there will be triple bonds. So if you want to open the two different strands of DNA, how many hydrogen bonds you have to cut here? 1, 2, 3, 4, 5, 6, 7, 8. Only 8 hydrogen bonds you have to cut here. Now if you want to open this particular piece of segment of DNA, how many hydrogen bonds you have to cut here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. You have to cut here near about 11 hydrogen bonds. So which is easier for you, to cut 8 hydrogen bonds or to cut 11 hydrogen bonds? It's obviously to cut 8 hydrogen bonds, and that is the reason for some plasmid easily you can open the replication site and they can replicate or produce their copy very rapidly. Okay. Now, uh, what are the three important features if you are choosing a plasmid as the cloning vector? Then what are the three important features you have to look inside? First, you have to look for the origin of replication. Just now I was discussing about the site of origin. What is the importance of origin? The origin site should be rich in A and T. There should not be G and more G and C. The origin site should contain more A and T double bond. Adenine always binds with 2 hydrogen bond with thymine. Okay. Now, origin is the site of initiation of DNA replication, and one more importance of origin or oricide you need to remember that it regulates the copy number of a plasmid. Okay, already we discussed that. Second thing, selectable marker or reporter genes should be present into the plasmid. Okay. Now, these selectable markers are called as reporter genes. Why? Because they helps in identification of the transformants and non-transformant. Like for example, if a plasmid is having these genes like AMPAR, ampicillin resistant gene, TETR, tetracycline resistant gene, or LAGZ gene, it will be very much useful for us to identify either our bacteria in lab in the culture is transformed with the recombinant DNA or not. So that's why we named them as selectable marker or reporter gene. Though most of the times we use antibiotic resistant gene, but sometime LAGZ gene is also used as reporter gene or selectable marker. Next, the third criteria you have to search for the cloning or the recognition site. Yeah, there should be presence of the palindromic sequence, whereby utilizing the restriction endonuclease you can produce sticky end cut and you can insert the gene of interest. Okay, so basically this cloning site or restriction sites are restriction sites for RAIN, means restriction sites for restriction endonuclease. Now, we will talk about first artificial plasmid that is known as PBR 332, and full form of PBR is plasmid of Bolivar and Rodriguez. Okay. So, scientists called Bolivar and Rodriguez, they constructed this particular artificial plasmid. Okay. Now, if you check this PBR 332, here first thing you are observing, that is presence of oricide. So, there are three criteria you have to fulfill: presence of oricide, presence of selectable marker, presence of cloning site. So, for PBR 332 we got the first criteria, presence of oricide. Okay. Next, you have to search for the second criteria, presence of selectable marker gene. Now, here we got two selectable marker gene. One is ampicillin resistant gene, another is tetracycline resistant gene. So, the second criteria that is also fulfilled. Now, you have to search for the third criteria, presence of cloning site or recognition site. Okay. Now, if you search inside the ampicillin resistant gene, here you can observe two different cloning sites of two different restriction enzymes. One cloning site for PVU1, another cloning site for PST1. If you check inside this ROP gene, that is called as repressor of primer gene, Inside there also you are getting cloning site of another restriction endonuclease PVU2. Inside this tetracycline resistant gene, you are again getting cloning site of two different restriction endonuclease BAMH1 and SAL1. And in between the zone of TET-R and AMPR, you are getting three cloning site of three different restriction enzymes, that is HIND3, CLA1 and EcoR1. So that's why in old days in genetic engineering to construct recombinant DNA technology, in the beginning they used to utilize PBR332 as cloning vector. Okay. So that was all about the PBR332. Now we'll talk about one important thing that is called as insertional inactivation. Okay. So what is insertional inactivation? Upon inserting the gene of interest in a cloning site within the antibiotic resistant gene. So if the cloning site is present within the selectable marker gene, then what will happen? The resistance against that particular antibiotic will be gone. If we talk about this PBR322, now if you want to input the gene of interest here is this site of PST1 restriction enzyme which is present inside AMPR gene. So what will happen? That will split the AMPR gene. You have to cut the AMPR gene. So the plasmid will lose resistance against ampicillin antibiotic. Similarly, if you want to insert the gene of interest into this site where you will be utilizing BAMH1 restriction enzyme. So here you have to cut this tetracycline resistant gene, you have to split tetracycline resistant gene to insert a new piece of DNA, and that what will happen then? The plasmid will lose the resistance against the particular antibiotic named tetracycline, and that is called insertional inactivation. Okay. So insertional inactivation can cause difficulties and complexity for some time. Like we know that these antibiotic resistant genes are acting as a reporter gene or selectable marker gene. AMPR or TETR, they are acting as a selectable marker gene or reporter gene. By treating this bacterial colony by ampicillin and tetracycline, we get the confirmation either the host bacteria which we are culturing to produce the product into our laboratory are transformed or not. But if they are inactivated upon insertion of gene of interest, then how to get the confirmation? How we will be getting the confirmation either your bacteria which is culturing for in your laboratory to obtain the product is originally transformed or not. So to avoid that complication, scientists they were searching for some alternative other than antibiotic resistant gene, and luckily they got another gene called LAG-Z. They got another gene called LAG-Z gene, and the plasmid was PUC19, plasmid of University of California. Okay, and 19 is the order number. Okay, so PUC19 plasmid, they constructed a plasmid called PUC19, and this PUC19 it had one ampicillin resistant gene and another selectable marker called as LAG-Z. Okay. Now, what is the function of LAG-Z? We already know the function of ampicillin resistant gene. What is the function of LAG-Z gene? lag Z will be producing a special enzyme called beta galactosidase, right? Now, in the culture medium, if you add chromogenic substrate as X-gal, this is the chromogenic substrate. So, enzymes need substrate. So, for this enzyme beta galactosidase, X-gal will act as a substrate. They will bind with each other, and as because the substrate will produce color upon binding, it will produce blue color. Okay. Now, this is for normal case. Now, what you will do? You will insert the gene of interest. You will insert the gene of interest inside the lag Z gene. Lag Z also contains the cloning site. So, you have to split lag Z to insert gene of interest. Now, your lag Z will lose the property of production of beta galactosidase. So, if you are transforming a bacteria by inserting this particular plasmid upon uh, when you want to check either the bacterial colony is transformed or not, you have to provide X-gal medium, X-gal, that is chromogenic substrate, into the medium. So, there will be no production of blue color, as because lag Z is already splitted by gene of interest, but you have this ampicillin resistant gene, you have this ampicillin resistant gene already intact, unsplitted into the another end of this plasmid. So, if you put ampicillin antibiotic and after that also this bacteria colony are not dying, not destroying, that means that bacterial colony is already transformed by taking the recombinant plasmid. Okay. So, what we have learned from this topic called insertional inactivation, previously in lab scientists used to use only the antibiotic resistant genes as selectable marker or reporter gene. But for some experiment they got complexities when they used to perform gene of interest insertion directly into the selectable marker gene like ampicillin R and tetracycline R. So, they were searching for alternative and they got another gene called lag Z. Now, lag Z is not responsible for resistance against any kind of antibiotic. Rather, lag Z in normal condition can produce blue color by producing beta galactosidase if you apply chromogenic substance into the culture medium. Now, if you insert the gene of interest inside the lag Z gene, there will be insertional inactivation of lag Z. Lag Z cannot produce beta galactosidase. Whenever you are putting X-gal into the medium, there will be no production of blue color, but as because already you have another selectable marker in that plasmid, that is ampicillin resistant or AMPAR, you can confer either the bacterial colony is transformed or not, or also by checking the color. If the bacterial colony unable to produce blue color, that is transformed. If the bacterial colony is able to produce blue color in the presence of the chromogenic substrate X-gal, that means that colony is non-transformant or not transformed. Okay. Now, what kind of vector or cloning vector can be used to transform animal cells? We have spoken about bacterial cell. We have spoken about how to transform dicot plants. For dicot plants, we can use Agrobacterium tumefaciens and the disarmed TI plasmid. For bacterial cells, if you want to transform it, you can use plasmid or you can use bacteriophage, disarmed bacteriophage. But for animal cell, you have to use disarmed retrovirus. What are retrovirus? They are primitive RNA virus, right? Like HIV, yesterday we were studying about HIV infection. So HIV is also one of the retrovirus. There are many retrovirus available. So what you have to do in laboratory, you have to exclude, you have to eliminate the disease causing potency of that particular retrovirus, and that is called as disarmed retrovirus. A retrovirus is normally responsible for causing disease for animal cell, but by applying biotechnology, you have to make it avirulent, you have to make it timid, you have to make it a good variety, a good strain, and then only it will be acting for you, it will be delivering gene of interest inside the animal cell. So that kind of retrovirus is called as disarmed retrovirus. With this retrovirus, you have to infect animal cell, but this infection will not cause a disease. Rather, the gene of interest will be transferred inside the animal genome. Okay. Now we have to know, we have to learn the art of transforming host cell. Okay. Already that uh, methods we were studying, they were about cloning vector or the transformation of host cell with the help of vector, or we can call those as vector mediated transformation. Now I will be talking about how to transform a cell without using a vector, non-vector mediated method. Okay. So if you want to transform a bacteria without utilizing a vector, then this heat shock method is used along with cold or chilled calcium chloride. Okay. So this chilled calcium chloride will make the cellular envelope more permeable or welcoming, more welcoming to the recombinant DNA or to the gene of interest. And by providing heat shock method, that means you will take the bacterial culture. Once you will be putting the bacterial culture at a temperature of 42 degrees Celsius in a medium where the surrounding is of 42 degrees Celsius. Again, you will be transferring the vial of bacteria inside ice, right. So there is sudden change of temperature, sudden change of drastic temperatures that will make the bacterial cell wall more porous and more viable, so that you can easily direct those bacterial cells, or you can make those bacterial cells competent to intake the gene of interest in a easier manner. Okay. But in normal condition, what happens is, because the cell membrane that is almost negatively charged and DNA is also negatively charged, so a cell is not very happy to intake the gene of interest or DNA inside. So that's why forcefully you have to put the DNA inside the cell by utilizing this heat shock and cold chilled calcium chloride tactics. Next, how to do for plants? Okay, Agrobacterium tumefaciens, they can only transform dicotyledonous plant. Now if you have to transform monocot, then you need to search for some other method. Now here we can go with this non-vector mediated transformation. You can use biolistic and gene gun. So by utilizing some microscopic guns, we will be transferring DNA or the recombinant DNA or gene of interest with the help of gold or tungsten particles by special process of bombardment, particle bombardment of gold and tungsten with the help of gene gun or biolistic. Okay. Now the next topic, the process of recombinant DNA technology. Okay. So sequence wise you have to follow them. First, you have to isolate the DNA. We have to learn the technique to isolate DNA from a cell. It's obviously we need to isolate the gene of interest. Okay, while you are taking out the DNA, isolating DNA from the donor cell, you have to cut the DNA into pieces. Then you have to amplify, you have to create millions of copies of that particular gene of interest, and then insertion of recombinant DNA into the host cell and obtaining the product, and lastly the downstream processing technique. Now let's check one by one how to isolate the gene from a host cell. Okay, now first you have to digest the cell wall and cell membrane. Now if it's bacteria, how to digest the cell wall? You have to use lysozyme enzyme to digest the cell wall of bacteria. If it's fungal cell wall, you can digest it by utilizing chitinase enzyme. If it's plant cell wall, you can digest it by utilizing cellulase enzyme. Next, what to do? Once the cell wall is digested by simple detergents or by uh, performing grinding technique, you can simply break down the cell membrane, right? Now you have to digest the impurities as protein and impurities as RNA, because you only want DNA, pure DNA. So how to clear those impurities? To get rid of proteins, you have to treat the solution with protease. Protease will be digesting these proteins present inside the cell, and RNase is one of the nuclease. They will be digesting the RNA present into that mixture. Okay, now your cell wall, cell membrane is gone, protein is gone, RNA is gone. So only pure DNA is collected and isolated now. And for the separation of the pure DNA, for the process of separation of the pure DNA from this mixture, what you have to use? You have to use chilled ethanol as written in NCERT. You have to use chilled ethanol, or chilled isopropyl alcohol can also be used, and that will be helping you to sediment the DNA strands into the mixture present into the test tube. By and the next thing what you have to do, by utilizing a sterilized iron rod or metallic rod or glass rod, you have to separate out the fibers of DNA from the mixture present in test tube, and the process is called as spooling. Okay. So what is the total process we are going to perform? When the DNA is isolated and separated, the next thing we have to do, we have to treat the DNA, isolated DNA, with REN, restriction endonuclease enzyme. Now, how this REN will be working? This restriction endonuclease will be cutting the DNA into smaller pieces, right? Now, you have to separate out the gene of interest. You need only the gene of interest, right? So how to do that? For that you have to run gel electrophoresis. Now you know that gel electrophoresis can separate out smaller pieces of DNA from larger piece. By the process of scooping and elution, we will be separating out and isolating out our gene of interest. Now here we got the desirable gene. Okay. Now next thing, you have to incorporate, you have to tag the gene of interest within a vector with a cloning vector or plasmid, right? Now when this desirable gene of interest is tagged with this cloning vector, this new molecule of DNA is called as rDNA or recombinant DNA. Okay. Now You have to run PCR. Why you need to run PCR now? Because the recombinant DNA is produced that is very low in quantity. So by utilizing some amplification technique or polymerase chain reaction, within within half hour or 1 hour, you can produce billions copies of these recombinant DNA, and simply now we'll be making the host cell competent and we'll be putting them inside the uh, system of host cell or the new alien cell. Okay, so let's check the process of polymerase chain reaction. Why it's called as polymerase chain reaction? Because here we be needing thermostable DNA polymer. We need to make multiple copies of that particular rDNA. So the process of making copies or to imitate the cellular replication mechanism into the laboratory. Inside our cell, how is cell is doubling the DNA by the process of DNA replication, right? Now, if you perform the similar kind of thing inside your laboratory by utilizing similar kind of enzymes, then the process will be called as polymerase chain reaction. But one thing is given here, you have to perform all these reactions, all this process in high temperature. That's why the machine that is used to perform these, that is called as thermal cycler. Thermal cycler is the name of the machine inside which we will be performing or amplifying the DNA. Okay. So first, at high temperature, see the temperature is near about 94 degrees Celsius temperature, you have to denature the double stranded DNA and you will be obtaining two anti-parallel separate strand. One is 5' to 3' and another is 3' to 5'. This first process is called as denaturation of DNA. When a double stranded DNA is separated into two single stranded one. Next, the process is called as annealing. Now, what do you mean by annealing? Again, you have to paste smaller pieces of genetic material as primer, that is called annealing. Annealing is done at the temperature of 55 degrees Celsius, and for annealing the smaller pieces of DNA you require, they are DNA primers. They are basically oligonucleotides. That means these are less than 10 base pair long. So this oligonucleotide DNA, single stranded DNA will be patched with the two different strands, the parallel strand and the anti-parallel strand, and now the annealing process is done. Now this short oligonucleotide pieces of DNA will be acting as primer, and you know that for the function of DNA polymerase you need these primers. Okay. Now the next step is to produce real double strand, and the step is called as elongation and extension, and for that you have to utilize DNA polymerase, and here you need thermostable DNA polymerase. That polymerase will not get destroyed at high temperature, and that is obtained from Thermus aquaticus, a thermophile bacteria, and the DNA polymerase of that Thermus aquaticus is named as TAC-Pol, Thermus aquaticus DNA polymerase. And you have to keep a lot of deoxyribonucleotides or DNTPs in the system, so that while this this TAC-Pol or the thermostable DNA polymerase is elongating the DNA strands, it can recruit these DNTPs to establish double stranded DNA from the template single strand, and that's how from one single sample of double stranded DNA two different molecules of double stranded DNA can be produced after one cycle. Now simply if you keep on running the cycle, after 30 cycles, near about 1 billion copies of double stranded DNA is produced, right? And this is the formula. N0 denotes the number of initial DNA and small n denotes the number of cycle. So the formula is N0, capital N0 into 2 to the power small n equals to copies of DNA produced. So as because the initial how many DNA strands we had taken, only one double stranded DNA was taken. So N0 will be 1 here, and how many cycle, suppose you have to perform 30 cycle. So what you have to multiply with 1, you have to multiply 2 to the power 30, and somehow you will be getting near about 1 billion copies of DNA. Now next process is obtaining the product and downstream processing. Now once successfully your cell is transformed, your host cell is transformed, you have to obtain the product, you have to culture the cell, inside large bioreactors in by maintaining some optimum pH, optimum moisture, optimum temperature, and that is the importance of utilizing bioreactors. And the process is simply called as tissue culture technique. Now why you need to perform tissue culture? You want, suppose you have transformed E. coli bacteria, by utilizing bioreactors you will be producing millions, billions cells. You want to multiply E. coli, those transformed E. coli cells, and when these E. coli cells are multiplied, and then it will be easier for you to extract the desirable product from them. Okay. Now if you talk about tissue culture technique, there are 3 modes of culture can be followed, right? One is continuous culture, another is batch culture, and another third one is fed batch culture. Now what is difference between continuous culture, batch culture, or fed batch culture? See, if you talk about batch culture is basically a closed system. Batch culture is basically a closed system. Culture media is given inside that tank, right? And you have to put some inoculum, means few bacteria we need to put inside that medium. So within few hours what will happen, this bacteria or these cells will be utilizing the nutrition present into the culture media and will multiply their number. But this batch culture, limit of this batch culture is very much limited, because after a certain point of time the cell number of cells are increasing inside this medium, and the resource, the nutrients are lacking gradually. They are exploiting the nutrients. So you keep that culture for a prolonged time. So gradually what will happen, first you will observe the number or the count is increasing, and when they have reached the saturation period, all the nutrients are finished, gradually the cells will start dying. So this kind of graph will be produced if you follow batch culture. That's why we won't be going with batch culture. Okay. So generally what we do, we perform continuous culture. Now, what do you mean by continuous culture? There are two outlets, two opening, one inlet and one outlet. With this inlet, there will be continuous supply of fresh culture media, fresh nutrients, and with the help of this outlet, gradually you are collecting out the expired medium or the excess amount of cells produced by your bacteria, or which is multiplied host cells inside it. So what will happen, there will be no saturation point. There will be no saturation point, and the graph that will be produced here like this. There will be log phase, there will be exponential phase, and there will be a stationary phase. There will be no lag phase, or there will be no downfall. That is the importance of continuous culture. You keep on adding culture media, fresh culture media, and you keep on eliminating the expired culture media and excess cells which are produced, which are growing inside. So, there will not be any sort of overpopulation of the host cell, of the culturing, the cell which you are culturing. If there is no overpopulation, the cells will remain healthy and you will get healthier, or means good product. Okay. So, this kind of bioreactors are utilized. These are some samples of different kinds of bioreactors used in factories. Now, the next step is downstream processing. So, downstream processing means DSP. Remember, downstream processing means separation and purification of the product. So, remember the continuous culture method that is happening inside the bioreactor. The culture is happening inside the bioreactor, and when you are done with the culture, you have to take out the culture media along with the cells and the product produced. So, you have to collect, right, and you have to send the sample for purification, because inside that liquid medium you have the remaining, the dead parts of cell and some impurities, right. So, you have to purify it, and then only you can store it and you can send the product to the market. So, the process of separation and purification is called as downstream processing. Now, if you talk about normal medicine, we consume in our daily life, right, when you fall sick. So, there are different brands available in market. For the same medicine, like for paracetamol, generally we take to reduce the fever. For one brand, they are selling one tablet is 50 paisa. Another brand is selling one tablet 1 rupees. Another brand is selling same paracetamol one tablet 2 rupees. Another brand is selling one tablet 5 rupees. So, what is the difference between that tablets? So, you will be finding all the tablets are containing 650 milligram of paracetamol inside that. So, everyone is giving you 650 milligram paracetamol inside that tablet. Now, why one tablet they are selling at the rate or price of 5 rupees per tablet, and why another company is selling at the rate of one tablet 1 rupees? So, the company which who is selling the same 650 mg of paracetamol in higher price, they have advanced technology, advanced downstream processing technique for the separation and purification of paracetamol from their culture, while they are processing it inside their laboratory. Okay. Now, if you check your NCERT, these two diagrams are given, and these are nothing but stride tank bioreactor. Okay, these are nothing but stride tank bioreactor. So, both are stride tank bioreactor, but the first one that is simple stride tank bioreactor, and the second one that is sparged stride tank bioreactor. Now, what do you mean by stride tank bioreactor? See, you are keeping culture media, inside the tanks, right, along with the growing cells, growing host cells, to obtain the product. Okay. Now, every time you need to shake the liquid, and that's why it said that stride tank bioreactor, they some either fans are present to shake the media, or always there is bubbling, formation of air bubble is there to shake the culture media. So whenever air bubbles are shaking the culture media is called as sparged tank bioreactor. When there is only fans, and propeller to shake the culture media, then that's called simple stride tank bioreactor. Now which is beneficial for what? If you are performing culture of aerobic organisms, then you must need to use purged stride tank bioreactor. Otherwise, after some time, the aerobic organisms, the aerobic cells will start dying inside simple stride tank. But if you are performing your downstream processing and the whole mechanism with some anaerobic cells, then you can go with simple or anaerobic bacteria culture. If you are doing anaerobic bacteria culture, you can go with simple stride tank. Okay, now within 10 minutes I will be starting the next chapter, that is biotechnology and its application. And up to this topic, if you have any question, if you want to ask something, you can write down your query into the chat section. Okay, so now start with the next chapter of this unit, that is biotechnology and its application. So, biotechnology has many applications such as biopharmaceuticals, that is in medicine, therapeutics, diagnostics, and genetically modified crops. We know about BT cotton, BT brinjal, processed food, bioremediation, waste treatment, and energy production. Okay, now this is very important uh, para here. Biotechnology has three critical research areas. What are they? They are providing best catalyst, means more purified or the best enzymes you are going to provide into any to perform a chemical reaction. More efficiency, more efficacy will be provided to that particular reaction, creating optimal conditions and downstream processing techniques. Okay, if you have more advanced downstream processing, you are going to produce superior product. Next, three options for increasing food production. Now, the areas where we need to explore biotechnology, first, that is agrochemical based agriculture. It uses fertilizers and pesticides, and for countries like India, it's quite expensive for normal farmers, and they will be causing environmental pollution if you use fertilizers, as because they are mostly inorganic in nature, and similarly pesticides, they are toxins. Okay, so they can cause soil pollution as well as water pollution. Now, another alternative of agriculture, that is called organic farming or organic agriculture. Again, for our farmers it quite expensive, and in the climatic condition like India, really you cannot think for pure organic farming. You have to take support of fertilizer as well as pesticide. So that's why we can think for the third option, we can explore genetically engineered crop or GMO, genetically modified organisms. So it uses genetically modified crops, and these genetically modified organisms are the plants, bacteria, fungi, animals whose genes are altered by manipulation. So here you don't need utilization of artificial or inorganic fertilizers as well as pesticides. Okay. Now we will try to compare between green revolution and the utilization of GMO. So, Green Revolution succeeded in tripling the food supply. So, before it was about quantity, right? So, Revolution is about quantity, Green Revolution is about hybridization and producing a superior variety by following the sexual mode of reproduction. Better management practices and agrochemicals are not affordable in developing countries. So, conventional breeding techniques are not efficient enough, as you know that the conventional hybridization technique will help you to get superior variety, but along with good genes, bad genes are also inherited to the next progeny. Okay, there is high chance of pollution is there if you use too much amount of agrochemicals, and sustainable agriculture was needed. Now, if you use fertilizer and pesticide, that is not sustainable form of agriculture. Eco-friendly, you need to use something which is eco-friendly. So, that is why genetically modified crops are possible solution. Again, you know that organic farming are expensive for those farmers. Okay. So, what is advantages of GM or genetically modified plants? So, it makes the crops more tolerant to abiotic stress like cold, drought, salt, heat or pH of soil. So, you will be getting a broad spectrum to culture the different varieties of plants. Pest resistant crops reduces the use of chemical pesticide, like example is BT cotton, and that is supporting sustainable agriculture, right, by reducing the pollutants in nature. It reduces post harvest losses, like for example flavor saver tomato. See, for potato, we can, after harvesting, we can store them for a few months or few years, even for few years, into warehouse, into cold storage. But it was previously it was not possible to store tomatoes into the cold storages. Okay. Now, with the help of genetic modification, we have produced a genetically modified variety of tomato called as flavor saver tomato. Now, this gene which was responsible for early rotting or early damage of these tomatoes is been inhibited by utilizing some rDNA, by following genetic engineering and applying rDNA technology. It increases the efficiency of mineral usage by plants. It prevents early exhaustion of soil fertility. Okay. That means you have to provide nutrients for the growth of plants, and most of the times the native plants, they are very much reluctant to absorb the nutrients or the organic nutrients, rather it can be said the humus from the soil. But the genetically modified plants are compelled to absorb those organic nutrients easily from soil. So, that is again beneficial for the farmer. It enhances nutritional value of food, that is example of golden rice. Golden rice is rich with vitamin A and iron. Golden rice is rich with vitamin A and iron. There was genetic modification done by combining the gene from daffodil flower plant and a bacteria called as Arwenia. So, scientists, they have taken genes from daffodil and Arwenia and incorporate inside the genome of rice, and that made a new variety called golden rice. To create tailor-made plants, means genetically modified plants are also known as tailor-made plants, to supply alternative resources like starches, fuels, pharmaceuticals, etc. To the industries. Okay. Now, if you use pest resistant plants like BT cotton, BT brinjal, they act as biopesticide. BT for Bacillus thuringiensis. If we talk about BT cotton, the BT toxin producing gene from Bacillus thuringiensis is introduced into the genome of cotton plant. That's why it reduces the need for insecticides or pesticide, and like BT cotton, we have produced BT corn, BT rice, BT tomato, BT potato, and genetically modified soybeans. Okay. Now we will be exploring that BT gene and the toxin produced by it. So here a disease of cotton ball is observed. Okay. So this is a cotton ball which is responsible in cotton plant for the production of cotton, and obviously if you are a farmer, you will be purifying this cotton, you will be collecting the cotton, and after purification it will be sent to the market. Okay. Now if these cotton balls are affected by cotton ball worms, if this cotton ball are affected by cotton ball worms, they are basically larvae of these moths, a special kind of moths. Now these larvae, they feed on the marrow or the ball kind of structure which is produced by the cotton plants, and when this cotton ball is destroyed or damaged, there will be no formation of cotton, and obviously that's economic harm for the farmer. Okay, that's why we have taken help from biotechnology. So we have observed that some strains of Bacillus thuringiensis have proteins that kill insects. Like, so what kind of insects can be killed by utilizing a toxin produced by Bacillus thuringiensis? Coleopterans like beetles, lepidopterans like tobacco birdworm or armyworm, basically they are kind of moths, and dipterans like different kind of flies and mosquitoes. So you need to remember this. The BT toxin produced by Bacillus thuringiensis bacteria, that is very much specific, that can only kill the larval stages of these insects, coleopterans, lepidopterans, and dipterans. Okay. So Bacillus thuringiensis forms an insecticidal protein called as BT toxin, which is crystal during a phase of their growth, and it does not kill the bacillus as it exists as inactive protoxin. Okay. So if you observe their growth phase, so a special phase of their growth, when these Bacillus thuringiensis, they are dying, then they start producing this particular crystal inside their cells, and these toxins are produced as inactive protoxin, inactivated form of toxin. So that's why these toxins are not harmful for those Bacillus bacterium, right? But when an insect ingests the toxin, it becomes active due to alkaline pH of their gut. Like, what is the alkalinity of our gut? 7.8, maximum 8.2 or 8.5, but the alkalinity of the gut of insects obviously more than 9. So if you are ingesting this BT toxin, you will not be dying. That's not harmful for us, but that's harmful for the larvae of those insects. So when the insect ingests, the toxin becomes active due to the alkaline pH of their gut, which solubilize the crystal, and the toxin binds to the surface of their midgut. You need to remember this. The toxin binds with their midgut epithelial cells and creating pores. Now if midgut is destroyed, you know that midgut of insects, that is the site of digestion and the absorption. So if there is no digestion, no complete digestion, no absorption of food, it will cause cell swelling and lysis, and obviously death of that insect, particularly the larval form. Okay. So these Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into crop plants such as cotton. So most Bt toxin are insect group specific. They are coded by cry genes. So you need to remember these Bt toxin producing genes are called as cry genes. Okay. Now proteins encoded by cry1AC and cry2AB, these genes will be controlling cotton bollworms, and protein that is coded by cry1AB, that will be controlling corn borer. Okay. Now if you want to learn the tricks of this chapter, how to remember all these things in an easier manner, you can check my one shot video which is already available into this channel. Okay, one shot of biotechnology and its application. You can check the video section. Okay. Now another practical instance of application of biotechnology, nematode resistance in tobacco plant, nematode resistance of tobacco plant. Okay. So basically tobacco plants, basically the roots of tobacco plants was getting destroyed by special kind of askelmint called as Meloidigyne incognita that infects the roots of tobacco plants, causing reducing in yield, and the disease is called as root knot disease. Okay, so this is the Meloidigyne incognita, a roundworm or askelmins that will be penetrating into the roots of tobacco plant and will be eating the tissue inside and will be destroying the productivity of the plant, and gradually the plant will die. Now, how to recover? For that again you have to take help of biotechnology. So it can be prevented by RNA interference or RNAi strategy. So what is RNA interference strategy? RNAi is a method of cellular defense in all eukaryotic organisms. It prevents the translation of a specific mRNA or silence that due to a complementary double-stranded RNA molecule. See what happens inside a cell. Suppose you got a particular harmful mRNA. This mRNA is harmful for you, right? This mRNA is the mRNA of the pathogen. So what this mRNA will be doing for? This mRNA will produce protein. This mRNA will produce protein, and this protein will cause harm to you. This protein will cause harm to you. Now, how RNA silencing or RNAi technique is done? Cell will be producing a double-stranded RNA. Generally, we know about double-stranded DNA, but cell has some special double-stranded RNA molecules, right? Now, when the cell will destroy one of the strand of double-stranded RNA, the remaining strand, that is called as antisense strand. Antisense strand. So that means the double-stranded RNA that was introduced to the cell, this strand is called as antisense strand. Now this sense strand can produce this sense strand is responsible for production of protein. This sense strand can be translated, but this antisense strand cannot produce protein, or the antisense strand cannot be translated. So that is the duty of RNAi technique, to produce antisense strand of RNA from a double-stranded RNA molecule. Now, what is the function of this antisense strand? This antisense strand will go and will bind with this mRNA of pathogen. This antisense strand will go and bind with the mRNA of pathogen, and once the antisense strand is binding with mRNA, protein synthesis process will be blocked, and if there is no protein formation or no pathogenic protein formation, there is no harm caused to the cell or to the host organ. So that's how it's written. That's why it's written here. RNAi method is a cellular defense in all eukaryotic organism. It prevents translation, protein synthesis of specific mRNA sequence due to, and how you are silencing, how you are stopping the translation of this mRNA with the help of a complementary double-stranded RNA molecule which is having one sense strand and another anti-sense strand. Now cell will be digesting the sense strand, remaining anti-sense strand will go and bind with this mRNA and will stop the process of translation. The source of this complementary RNA is from an infection by RNA virus or mobile genetic elements or transposition that replicate via an RNA intermediate. Now, if you ask, sir, how the cell will be getting this double-stranded RNA? The answer is given here. The source of this complementary RNA is from an previous infection. The double-stranded RNA was produced inside the cell as a result of some previous infection by RNA virus or some mobile genetic elements or transposition that replicate via RNA intermediate. Okay, again this transposition are also infecting elements, infecting RNA components. When a cell is affected by RNA virus, when a cell is affected by retrovirus, then your cell can get this kind of vagabond pieces of RNA. We call them transposition, and sometimes this transposition can act as the source of that complementary double-stranded RNA, which is rather later on can be required to perform RNAi technique. Now, for our case, what we have to do? We have to isolate that nematode specific genes. It is introduced into the host plant using agrobacterium vectors, and it produces both sense and antisense strand in host cell. Now the RNA are complementary, so they form double-stranded double-stranded RNA, and it initiates RNAi and silences the specific mRNA of nematode. Thus, the parasite cannot survive in the transgenic host expressing specific interfering RNA. Now, how this Meloidigine incognitia is stopped? Now, suppose this is the Meloidigine incognitia. Suppose this is the pathogen. This is Meloidigine incognitia, and that's very hungry, that's feeding on tobacco plant. And whenever it will be feeding upon the tissue from the tobacco root, it's gaining the nutrients. A special protein is produced. A special protein is produced from mRNA into its body. Whenever it is eating or feeding on the tissue of tobacco plant, the cells of this Meloidigine incognitia producing a special mRNA. When the mRNA is translated into some protein, and this protein is helping the nematode, helping the roundworm to become more powerful and more strong. Okay. Now you have to stop this process. Now what to do and how to do. So this is the tobacco plant cell. So what we have to do, we have to construct a DNA molecule. We have to construct a DNA molecule uh, which can code for, we have to construct a DNA molecule which can code for this particular mRNA of this Meloidigyne incognitia, and we will be inputting that into the tobacco plant cell, and by the reverse transcription, by following a special technology called as reverse transcription technology, these double stranded RNA is converted into double stranded DNA, right, inside the cell. Now this double stranded DNA will produce mRNA by the process of transcription. By utilizing the same process, by the technology is called as antisense technology. One antisense strand of mRNA is produced, that is called as antisense RNA. So inside the cell, two forms of RNAs are produced, normal mRNA and antisense mRNA. Now this normal mRNA and antisense RNA, means sense RNA and antisense RNA, they are complementary to each other. They are complementary to each other, so they will be binding to form a double stranded RNA molecule. Now this double stranded RNA molecule will be cleaved by a special complex of protein called as RISC protein and dicer protein. These are some enzymes which are responsible for cleaving the sense mRNA. The sense mRNA will be cut into smaller pieces. The sense mRNA will be cut into smaller pieces, but there will be intact antisense mRNA still remaining inside the cell. So the antisense RNA is still remaining inside the cell. Now this is called genetically modified tobacco plant cell. It is having one molecule of antisense mRNA inside this. Now this Meloidigyne will be feeding on this particular tobacco plant. While it's feeding, it will get the antisense RNA. While it's feeding or digesting this genetically modified tobacco plant cell, it will be getting this antisense RNA into its body, and this antisense RNA will go and bind with the protein producing mRNA, and that will block formation of protein into the body of Meloidigyne, and if protein is not formed, it will not be getting nutrition, and if it's devoid of nutrition, it will die or it will reduce its attack towards the tobacco plant. Now we'll talk about application of medicine, application of biotechnology in medicine. So products from non-human sources cause unwanted immunological responses but recombinant therapeutics does not have such problems. It's like that for insulin. Insulin injection is taken if someone is suffering from diabetes mellitus, right? But previously, the insulin, the people used to collect the pharmaceutical industry used to isolate insulin from pancreas of cattle, from pancreas of goat, horse, or some other cattle. Now those animal insulin used to injected for human, used to produced for human, and we used to inject those animal produced insulin inside our bloodstream, and they used to cause allergic reaction or immunological response inside our body, so that genetically modified organisms were prepared, genetically modified E. coli bacteria were prepared by one company called as Eli Lilly, and for the first time successfully they prepared human insulin in lab condition. Okay. So it said that at present about 30 recombinant therapeutics have been approved, and of these 12 are being marketed in India. Now let's talk about the example of formation of genetically engineered insulin. So insulin is used to manage adult onset diabetes or diabetes mellitus, right? That is also called as NIDDM, non-insulin dependent diabetes. And this insulin from the pancreas of animals like cattle or pigs causes allergy. But now it is possible to produce human insulin using bacteria, genetically modified bacteria. Yes. So insulin consists of two short polypeptide chain, that is chain A and chain B. They are connected by disulfide bridges. Okay. So this is the activated form of our insulin. This is A peptide, this is B peptide, and they are connected by disulfide bonds. They are connected by disulfide bonds. But inside our pancreas, initially the insulin is produced, that is called as proinsulin, that is inactivated format. Now by the process of maturation, it's our biosynthetic mechanism inside our cell which will be cleaving the extra polypeptide chain called as C chain. Now the proinsulin is having three protein chain, three polypeptide chain, chain A, chain B and chain C. Now which is causing disturbance to us? Chain C. Our body will cleave chain C and separate it out, and that will create active form of insulin. Okay. Now in mammals, insulin is synthesized as prohormone called as proinsulin. It is processed to become mature and functional hormone inside our body, and that prohormone contains an extra stretch called C peptide, and this is removed during the time of maturation stage. So this step is called as maturation of insulin. Maturation of insulin, when active form of insulin is produced inside our body from proinsulin or inactivated insulin. Now in the year of 1983, Eli Lilly, an American company, prepared two DNA sequence corresponding to A chain and B chain. Okay. Now what is the problem? This A chain, B chain and C chain, this proinsulin is produced inside our body from a gene. Insulin producing gene is responsible for the production of proinsulin into our body, and that is a single entity. That gene is a single entity. Now if you are performing normal genetic modification, if you have isolated insulin producing gene from human cell, and if you incorporate by following the rDNA technology inside the E. coli bacteria, that can produce pro-insulin, that cannot produce active form of insulin, and it is not possible for us to modify a pro-insulin molecule into active form or functional form of insulin by cleaving the C peptide. In laboratory, that is not possible for us, right? Or that is a very much hideous job, very tough job. So what Eli Lilly company, scientist of Eli Lilly, what they did, they prepared two different gene sequence corresponding. One can produce the protein of polypeptide A, and another gene which can produce the polypeptide chain of B of human insulin. And for these two different genes were introduced into plasmid of E. coli bacteria to produce insulin chains. So chain A and chain B were then combined by creating disulfide bonds to form human insulin or humalin. So what was the problem? If you are collecting human insulin, if you are collecting human insulin gene, if you are collecting human insulin gene and putting it into a vector, that human insulin gene, if you are putting into a vector forming rDNA, and then if you put the vector, insert the vector inside E. coli bacteria, then what will happen? Then simply the gene will survive inside the E. coli, and the E. coli will produce insulin, but the inactive form, that is called as proinsulin, that is non-functional in nature. So that what if you perform the normal rDNA technology, and there is no chance or there is no method known to us, you can convert this proinsulin into functional or matured insulin. So for that, what we have to do, we have to create a sequence of two different DNA. We have to construct sequence of two different DNA. One can produce A peptide, another can produce B peptide, right? Now if you incorporate these two genes into two different E. coli bacteria, with the help of plasmid, A peptide gene and B peptide gene, this is containing A peptide gene, this is containing B peptide gene. This E. coli bacteria will produce A peptide for you, this E. coli bacteria will produce B peptide for you, and later on by constructing a disulfide bond, you can merge these A peptide and B peptide, and thus functional insulin is produced. Okay. Next, another application of biotechnology, that is gene therapy. Now gene therapy is a method to correct a gene defect in a child or embryo. Here genes are inserted into a person's cells and tissue to treat a hereditary disease. It compensates for the non-functional gene. So most of the time we can cure inherited or congenital diseases with the help of gene therapy. Okay. Now, first clinical gene therapy was performed in 1990, was given to 4 years old girl child who was suffering from adenosine deaminase deficiency. Okay. Now, this adenosine deaminase is responsible for maturation of T cell of our body, right? So, if your body is unable to produce adenosine deaminase, there will not be any kind of maturation of T cell into our body. If T cells are not matured, your immune system will become very much weak. Okay. So, this is caused due to the deletion of the gene of adenosine deaminase, an enzyme for the functioning of immune system, and it can be cured by bone marrow transplantation or by enzyme replacement theory, that is injection of adenosine deaminase is given. But none of the process are not completely curative. If you transplant the bone marrow, again within few months you will be facing that disease. If you inject adenosine deaminase enzyme, there is very less probabilities there that will act properly for your body. So, that's why we won't go with these two processes. We won't be going with bone marrow transplantation, or we won't be going with enzyme replacement therapy. Rather, what we can do, we have to go for direct gene therapy for this particular deficiency. So, for that what you have to do, you have to collect lymphocyte from the patient's blood. You have to grow in a culture medium, and to those cultured lymphocyte, to those cultured T cells or B cells, you can introduce the functional ADA complementary DNA. ADA complementary DNA into the lymphocyte using a retroviral vector. So, here these lymphocytes are transformed with the help of disarmed virus or disarmed retrovirus. They are returned to the patient's body, and this should be periodically repeated, as lymphocytes are not immortal. Within few days, those lymphocytes will die inside the body, and again a new set of genetically modified lymphocytes are inserted into the body. Okay. So, if ADA gene from marrow cells is introduced into cells at early embryonic stages, it could be a permanent cure. But for her, that 4-year-old child, it was not a permanent cure, but till then it was working for her, the process was working for her betterment. So, basically what we have to do in that case, we have to take out T lymphocytes, the defective T lymphocytes from the bloodstream, from the bone marrow of that child. We have to culture into the animal culture media to increase their number into millions. Then what we have to do, then we have to insert the complementary DNA. We have to insert the DNA which is having capability to produce adenosine deaminase enzyme, which is having capability to produce adenosine deaminase enzyme into those lymphocyte cells. Now, when you are performing recombinant DNA technology, these default or these defective lymphocytes are will be acting as good lymphocyte or functional lymphocyte. Now, with the uh, following the proper technique, you have to inject this lymphocyte into the body of that girl. Now, for few months or for few days, this transformed lymphocyte, this transformed lymphocyte inside the body will produce ADA enzyme, adenosine deaminase enzyme, and that will provide immunity to that child. And after some time, after few days, again you have to take lymphocyte from her body and you have to perform the same thing and you have to keep on doing it again and again. And if you want permanent cure, then you have during the time of embryonic developmental phase of that child, you know, there are techniques, uh, you can measure or you can get to know either that fetus growing inside the mother's womb is having any sort of genetic disease or not. So if you got to know that fetus is having suffering from ADA deficiency in the embryonic phase only, if you replace with genetically modified stem cells, then only there is a possibility that you can crack a permanent cure of this particular disease. Okay. Now we will go with next topic, that is molecular diagnosis. For conventional methods, diagnosis means how to determine either a person is suffering from a disease or not. So there are some conventional method like pathological test of serum, urine, saliva are not suitable for early diagnosis of disease. At least you need few days to get the result, to get the reports. Okay. It is possible by the techniques such as recombinant DNA technology like PCR, ELISA. Within few minutes you can get the result. Like if you remember the days of COVID, when you used to travel from one place to another place, okay, into the airports, into the railway junctions or railway platforms, within just 10 minutes by performing RT-PCR, they used to confirm either the passenger is COVID positive or COVID negative. So, if we talk about RT-PCR, reverse transcriptase PCR, one of the technique of molecular diagnosis to confirm either a person is having the person is affected by COVID virus or not. Okay. So first thing is polymerase chain reaction PCR. Already we have discussed about that. If you have very low concentration of bacteria or virus, can be detected by amplification of their nucleic acid by PCR. So if I collect saliva sample from your body and inside the saliva sample, suppose there is very low titer, very low concentration of the viral DNA or the bacterial DNA is present. So if we perform PCR within few minutes or within half an hour, we just have observed from one single DNA you can create near about 1 billion copies of sample. Okay. Now, where you can use PCR? To detect HIV in suspected patients, to detect gene mutations in suspected cancer patient, to identify many other genetic disorder. So, if you are infected by retrovirus, if you are observing malignant tumor formation inside your body, or if you are suffering from any other genetic disorder, the change of DNA, okay, the malfunctioning of the DNA can be detected by DNA amplification or the PCR technique. Okay. Next, you also can go with ELISA. The full form is enzyme linked immunosorbent assay. So what is done in case of ELISA? It is based on antigen antibody interaction infection by pathogen can be detected by presence of antigens, that special proteins or glycoproteins, or by detecting the antibodies synthesized against the pathogen. So it's very simple. You have to perform this ELISA test. You have to search for presence of antigen or antibody into the body fluid of the person. Now, if you are suffering from, suppose you are suffering from dengue, then obviously you are infected with dengue virus, right? When this dengue virus will enter into your body, they will start producing toxin, and that's why you are suffering. Now those toxins produced by the dengue virus or den virus, they are actually the antigens. Okay. Now if I take out your body fluid sample from you, and if I put inside the small vial, right, where I will be performing this ELISA, and what I have to do to confirm that, I have to add antibodies which can confirm me, which can confirm that pathologist, that if there is reaction happening in between that antigen, dengue antigen and dengue antibody, that means if there is binding, obviously the patient is suffering from that particular disease, right. Now how that pathologist will get the confirmation? How he or she will know that inside that vial the liquid is there, the antigen and antibody is binding with each other? So a signal is required, and for that signal you need to utilize chromogenic or a special enzyme labeled antibodies. When those enzyme labeled antibodies will bind with the specific antigens, you have some chromogenic substrate present into that vial, they will produce special color. And by observing formation of color, the color formation denotes antigen is binding successfully with antibodies. That means presence of disease causing element into your body fluid. Now we will talk about transgenic animals. So these are the animals whose genome has been altered by introducing of a foreign gene by manipulation. Like transgenic rats, we have produced transgenic rats, rabbits, transgenic pigs, sheep, cows and fish, and you need to remember this, over 95% of the transgenic animals are mouse. Now what kind of uh, welfare activities can be done with these transgenic animals? What are the benefits? To study the regulation of genes and their action on normal physiology and development, that is study of insulin like growth factors. Whenever you are researching into your laboratory, you cannot check with human being. Suppose if you want to check the progression of a disease inside animal, so you cannot ask me or the scientist, they cannot ask you, please come into my laboratory, I want to treat you as a means experiment model, right. So in that case the transgenic animals can be used to study the contribution of genes into the development of disease, and thereby new treatments like transgenic models for human disease such as cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's disease. Biological products like human protein called alpha 1 antitrypsin used to treat emphysema. In 1997, Rosie, the first transgenic cow, produced human protein enriched milk, that is called as alpha lactalbumin. Vaccine safety testing is also done. Transgenic mice are used to test the safety of the polio vaccine. If it is reliable, they can replace the use of monkeys. So when uh, in pathological laboratories or uh, scientists, they are trying to produce vaccine against a disease, first they will do a dry run, right? They will take model organism as animals, these rats or mice, and then they will be injecting that vaccine, and we will be checking either that is causing any threat or that is causing any side effect inside the body or not. Generally, at a successful lab trial, they will be injecting the vaccine in monkeys, in primates, is because monkeys will be sharing near about 99% of genes similar with us. So instead of applying or utilizing human being as a model, scientists as their trial method, they uses they will be using these rats or mice or the monkeys. Okay. So that is called as vaccine safety testing, because for the first time, if today if I make a vaccine against any disease, suppose there is a recently there was a outbreak of Nipah virus, right? Recently there was an outbreak of another disease called as Nipah virus. Now suppose you are the scientist, you are researching in your lab and you are trying to prepare a vaccine against that. Now like according to you, you thought that you have prepared that vaccine into your laboratory. Now you want to uh, check some trial. Is it really working for human being, or can it cause any harm when we are injecting into our body? So directly you cannot inject or you cannot try upon human being. Before that you have to cross through or you have to pass some trials on animals, and if it's successfully passed, then only you will be subjected to try your vaccine or newly modified vaccine or newly produced vaccine upon a particular community of men. Then if we talk about chemical safety testing, toxicity testing, some transgenic animals carry genes which make them more sensitive or toxic substance than non-transgenic animals. So they are exposed to the toxic substances and the effect is studied, and it will be giving us immediate result. Okay. So these are some examples of transgenic animals. So this is fluorescence, these rats or these mice are showing fluorescence, and how they were produced. The light emitting gene or the fluorescence producing gene from jellyfish was incorporated into the genome of these mice. This is Rosie, and already we have studied about the Rosie, the transgenic cow. It produced human protein, milk, and that milk was enriched with human alpha lactalbumin, and you have to remember every liter of her milk was containing 2.4 gram of human alpha lactalbumin. Okay. This is Andy, another transgenic animal. Basically, it's a transgenic monkey. Again, we did the same thing. We have isolated fluorescence or light emitting gene from jellyfish and inserted into the cells of this particular creature. Okay. Now we need to talk about some ethical issues. Okay. So problem of unpredictable result. First, genetic modification may cause unpredictable result, like Indian government has set up organization like GEAC, Genetic Engineering Approval Committee, to make decisions about the validity of genetic modified research and the safety of GMO for public service. Suppose tomorrow you are coming with a superior variety of a crop and you said that I have prepared a superior variety of rice which is rich in vitamin A, vitamin B, vitamin C, vitamin D, all the vitamins are there, and you are going to get a lot of minerals into that, and you have produced that by following rDNA technology. Now first your thesis or research will be sent to GEAC. First they will check the credibility of your invention, or whatever you have prepared, the superior variety of the superior crop. Then you have to face a trial method, okay, and upon successful completion of the trial of your superior variety, whatever you have produced into your lab, then only it you will be eligible to send or trade into market. Now what is biopiracy? It is the use of bioresource by multinational companies and other organizations without proper authorization from the countries and people concerned. Like basmati rice has a unique aroma and flavor, and India has 27 varieties of basmati. In 1997, an American company called Rice Tea got patent rights on basmati rice through the US Patent and Trademark Office. Now this allowed the company to sell a new variety of basmati rice. Now this was actually derived from Indian farmers' varieties, right? Now Indian basmati was crossed with a semi-dwarf variety and claimed as a novelty. So other people selling basmati rice could be restricted by patent. Now as because they have patented that hybrid variety of basmati rice. Now as a native farmer, suppose you have some native varieties of basmati and you want to sell into the market of USA. So what you have to do, you have to give royalty to that company if you want to sell your native variety of basmati rice into the market of USA. But originally what they did, they have done piracy. They had taken your technology, they had learned how you are preparing basmati rice into this climate, and they have crossbreeded with another dwarf variety, and they just have produced a hybrid one. So that is not called novelty, that is not called uniqueness. So that kind of things are quite problematic when you are talking about biopiracy. Okay, as because that company, that authority is much powerful than you, they are just trying to exploit, nothing. Some herbal medicines like turmeric and neem are also some examples. Yeah, few British companies, they were trying to take patent for this particular Indian herbs, and we know that Indian Ayurveda system is utilizing turmeric, neem, and other herbs from thousands of years ago. Now, Indian Parliament has cleared the second amendment of the Indian Patent Bill, and that has considered patent terms emergency provision and research and development of initiatives. Now, in order to stop that unscrupulous practice by some other countries or some powerful people, Indian government has patented that bill, okay, in order to stop those malpractices. So with this we are done with the units of biomolecules.